Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the grand tapestry of 20th century science, certain publications serve as singular inflection points, moments when the cumulative weight of disparate discoveries coalesces into a new and powerful synthesis. The 1972 paper from the laboratories of Stanley Cohen and Herbert Boyer, titled Construction of Biologically Functional Bacterial Plasmids in Vitro, represents such a watershed. Authored by Cohen, Chang, Boyer, Robert Heyneker and Herbert Heyneker, this work, published in the Proceedings of the National Academy of Sciences, possesses a deceptive simplicity in its methodology that belies its revolutionary implications. It detailed a procedure for the deliberate and precise combination of genetic material from different sources into a single, replicable molecule. This was the formal inauguration of genetic engineering, a moment that transformed biology from a science of observation and analysis into one of composition and creation, with consequences that continue to reverberate through every facet of modern life science. The paper was the culmination of decades of foundational research in microbial genetics and biochemistry, a perfectly timed convergence of tool and concept that unlocked the ability to rewrite the very script of life. The intellectual landscape of the early 1970s was fertile ground for such a breakthrough. The essential components, like instruments in an unassembled orchestra, lay scattered across various subdisciplines, awaiting a conductor. The first of these was the bacterial plasmid, whose existence was brought to light by Joshua Lederberg's work in the 1950s. These small, circular, extrachromosomal pieces of DNA were initially seen as curiosities, agents of a peculiar form of bacterial promiscuity responsible for transferring traits like antibiotic resistance between cells. Stanley Cohen had dedicated his research to understanding the biology of these plasmids, particularly one he isolated and named PSC101, which carried a gene conferring resistance to the antibiotic tetracycline. He had developed robust methods for isolating these plasmids and, crucially, for reintroducing them into susceptible Escherichia coli cells, a process known as transformation, and confirming their functional uptake. The plasmid, with its ability to replicate independently within a host bacterium, presented itself as a potential vehicle, a molecular ferry for carrying foreign genetic cargo. The second indispensable tool emerged from a seemingly unrelated line of inquiry into how bacteria defend themselves against invading viruses. The work of Werner Aber, Hamilton Smith, and Daniel Nathans revealed the existence of restriction enzymes, proteins that act as molecular scalpels. These enzymes recognize specific, short sequences of DNA and cleave the double helix at those precise locations. Herbert Boyer's laboratory had isolated and characterized one such enzyme, EcoR1, from a strain of Escherichia coli. EcoR1 was exceptionally useful because it made a staggered cut in the DNA backbone at its recognition site, the nucleotide sequence guanine adenine adenine thymine thymine cytosine. This produced fragments with short, single-stranded overhangs. Because the overhangs of any two DNA fragments cut by EcoR1 are complementary, they can readily anneal to each other through hydrogen bonding, creating what the researchers aptly termed sticky ends. The final piece of the molecular construction kit was the enzyme DNA ligase, isolated in the 1960s by several groups, which could form the strong phosphodiester bonds needed to permanently seal the nicks in the DNA backbone after two fragments had been joined. The plasmid was the vector, the restriction enzyme was the scissors, and the ligase was the glue. All the necessary elements for molecular carpentry were finally on the laboratory bench. The 1972 experiment itself was a masterpiece of elegant logic and execution. The researchers took two separate plasmids. One was Cohen's well-characterized PSC101, carrying its tetracycline resistance gene. The other was a larger plasmid, R6-5, which conferred resistance to multiple antibiotics. Their objective was to cut pieces from these plasmids and stitch them together to form a novel, man-made plasmid, and then to prove that this new creation was biologically active within a living cell. Using the EcoR1 enzyme, they cleaved both types of plasmids into a collection of DNA fragments. Because EcoR1 cuts at specific sites, this process yielded a defined set of fragments for each plasmid. They then mixed these fragments together in a test tube. The complementary sticky ends, generated by the EcoR1 cuts, naturally found each other and paired up, forming various combinations of cobbled-together DNA. Some fragments would simply rejoin with their original partners, while others would anneal with fragments from the other type of plasmid, creating chimeric molecules. Following this annealing step, the researchers added DNA ligase to permanently weld the fragments into stable, circular plasmids. The result was a heterogeneous soup of DNA molecules reconstituted original plasmids and, crucially, new hybrid plasmids containing genetic information from both PESE1 and R6-5. The genius of the experiment lay in its subsequent selection process. The researchers introduced this plasmid mixture into a strain of Escherichia coli that was naturally susceptible to tetracycline. They then plated the transformed bacteria onto a growth medium containing tetracycline. Only those bacteria that had successfully incorporated a plasmid carrying the tetracycline resistance gene from PESE101 could survive and form colonies. By analyzing the plasmids from the surviving colonies, Cohen, Chang, and their colleagues demonstrated that they had indeed created new, functional molecules. They had isolated plasmids containing fragments from both parental molecules, proving that a composite plasmid, constructed entirely in vitro, could be introduced into a host cell where it would replicate and express its genetic information. The bacteria were reading a sentence of genetic code that had been written by human hands. The publication of these findings sent an immediate and powerful shockwave through the biological sciences community. The implications were clear and breathtaking. The technique offered a universal method for isolating any gene of interest, inserting it into a plasmid, and then producing it in vast quantities within rapidly dividing bacteria. Biology had acquired its own version of the integrated circuit. The commercial potential was likewise instantly apparent. Herbert Boyer was famously approached by the young venture capitalist Robert Swanson, a meeting that led to the founding of Genentech in 1976, the world's first biotechnology company. The explicit goal was to harness this new recombinant DNA technology to produce medically important human proteins. Their first landmark success, the synthesis of human insulin in Escherichia coli, was achieved in 1978 for millions of diabetics who previously relied on insulin extracted from pigs and cows, which could cause allergic reactions. This was a medical miracle. It was followed by the production of human growth hormone, interferons, and a host of other therapeutic proteins. The 1972 paper had moved from a proof of concept to a life-saving industrial process in less than a decade, a velocity of translation from basic science to application but has few parallels. The ability to manipulate genes with such precision catalyzed a gold rush of discovery and innovation, laying the economic and scientific foundation for the entire biotechnology industry. With this Promethean power to reshape living organisms came profound and unsettling questions. The creators of this technology were among the first to recognize its potential dangers. What if a harmless bacterium like Escherichia coli were accidentally endowed with genes for toxin production or high-level antibiotic resistance, creating a novel pathogen that could escape the lab? This deep sense of responsibility among the leading scientists in the field led to an unprecedented event in the history of science. In 1974, a group of prominent molecular biologists, led by Paul Berg, published a letter calling for a voluntary, temporary moratorium on certain types of recombinant DNA experiments, particularly those involving genes from cancer-causing viruses or those that could increase the virulence of pathogens. This call for a pause culminated in the landmark Asilma Conference on Recombinant DNA in February 1975. At this meeting, 140 scientists, along with lawyers and journalists, convened to debate the risks and to forge a consensus on how to proceed safely. Cohen and Boyer were central figures in these discussions. The outcome of Asilma was a set of principles for conducting recombinant DNA research based on the level of perceived risk, recommending specific physical and biological containment measures for different classes of experiments. These principles formed the basis for the formal guidelines later issued by the National Institutes of Health in the United States and adopted by regulatory bodies worldwide. The Asilma Conference stands as a remarkable testament to scientific self-governance, a moment when the community of researchers collectively grappled with the ethical dimensions of their own work and chose a path of public accountability and cautious progress. It established a vital precedent for the governance of powerful new technologies that would follow, from human genomics to gene editing. The long-term legacy of the Cohen and Boyer paper is, quite simply, the fabric of modern biology. Recombinant DNA technology became a fundamental, routine tool, as indispensable to the molecular biologist as a microscope. It powered the great waves of discovery that followed. The ability to clone and sequence genes made massive undertakings like the Human Genome Project conceivable. It enabled the creation of transgenic organisms, leading to genetically modified crops with enhanced yield and nutritional value, and laboratory animals engineered to model human diseases, accelerating the search for cures. The core principle of cutting and pasting DNA has evolved, giving rise to ever more sophisticated techniques. Modern gene editing systems like CRISPR, Cas9, while mechanistically different, are the direct intellectual descendants of the foundational work done in 1972. They operate on the same paradigm: the targeted modification of a genetic sequence to achieve a desired biological outcome. from personalized medicine and gene therapy to synthetic biology and industrial fermentation, there is scarcely an area of life science that remains untouched by the methods and the mindset inaugurated by the construction of the first biologically functional plasmid in vitro. In conclusion, the 1972 paper by Cohen, Chang, Boyer, Helling and Heyneker is far more than a classic of the scientific literature. It is a charter document for a new era of biological science. By masterfully combining insights from plasmid genetics with the new tools of enzymology, the authors provided a simple, robust, and universally applicable method for engineering life at its most fundamental level. The work solved a technical problem, but in doing so, it dissolved the conceptual barrier. It demonstrated that the genetic code was a fluid and manipulable medium, its components from disparate branches of the tree of life capable of being united into novel, functional wholes. This realization not only launched a multi-billion-dollar industry and revolutionized medicine, but also forced a necessary and enduring conversation about the ethical responsibilities that accompany the power to create. The construction of a single hybrid plasmid in a test tube was a small technical step that signified a giant leap in human capability, forever altering our relationship with the natural world and placing a future of evolution, in some small measure, into our own hands.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.